Welcome back to Invisible Worlds. Today we are diving into a groundbreaking discovery in the science of aging. Before we jump in, just a quick reminder, this channel is all about real science. We break down fascinating discoveries in biology, biotech and beyond, purely for curiosity and learning. So today's topic is about aging, but we are not talking about miracle pills or anti-aging hacks. This isn't about reversing time with a magic drug. What we are discussing is a fascinating piece of research recently published in one of the world's most reputed scientific journals. If you're curious about the weird, wonderful, and invisible things happening inside your body, you're in the right place. So let's dive in. In a recent study, scientists uncovered a breakthrough, a map of longevity associated acetylation sites in mammals. It might sound like an alien language, but it is pretty simple. Acetylation is a process in our cells by which tiny chemical marks, which we can call acetyl groups, are added to certain locations in our proteins. And why is this important? The addition or removal of acetyl groups can change how proteins behave. Think of them as molecular switches. Adding an acetyl group can turn a protein on or off, change its shape, or even control where it goes within the cell. Check out our protein basics video in the top right corner if you want a quick refresher. As we age, our bodies start to wear down. Cells don't divide as well, DNA gets damaged, and age-related diseases begin to take over. But some animals live much longer than others. It turns out the secret isn't just in their DNA. It is in how their proteins are modified through acetylation. So what did they do in this study? The scientists used a powerful tool called Faro to compare proteins across 107 different mammals. They chose mammals from tiny mice to massive whales, and they identified over 482 acetylation sites in mice and 695 in humans that are directly linked to longevity. Here is where it gets fascinating. In long-lived animals, these acetylation sites are often locked in place, making proteins more stable, protecting them from damage, or boosting their beneficial functions. For instance, one of these proteins, CBS, plays a critical role in producing a gas called hydrogen sulfide, which protects cells. Long-lived animals have a specific acetylation pattern on CBS that makes it produce even more H2S, extending their lifespan. But how do these acetylation sites actually work? This study revealed two main types of longevity-associated acetylation. First is acetylation gain. At a particular location in a protein, short-lived animals have the amino acid lysine. In the long-lived animals, the same lysine is acetylated, meaning an acetyl group is added. This can activate or inactivate important proteins. The second is acetylation loss. In the second type, long-lived animals replace the acetylated lysine with amino acids like arginine or glutamine. So what does glutamine do? Glutamine makes the particular location permanently acetylated, and conversely, arginine makes it permanently deacetylated. So this is a way of locking the protein in a state that helps in longevity. Now, the question is, which are the proteins that are modified this way? Are these changes random? As with anything concerning nature, these are not random. They are precisely engineered to target proteins involved in DNA repair, cellular energy production, and even anti-cancer mechanisms. Why does this matter? Because these changes are not just about living longer, they are about living healthier. In fact, the study found that proteins involved in DNA repair were especially impacted by acetylation. One example is USP10, protein that is involved in DNA repair. In short-lived animals, it is deacetylated. But in long-lived species, it acquires an acetylation site, and that acetylation helps prevent cancer. This means long-lived animals are not just lucky, they are built to resist the damage that comes with age. So what next? Scientists are now exploring how to use these acetylation switches to boost human health and lifespan. Can we replicate this in humans? Can we activate these protective marks to fight age-related diseases? The science is still young, but one thing is clear. Understanding acetylation could open the door to a longer, healthier life for all of us. Thank you so much for hanging out with us today. Until next time, stay curious.